Howdy, everyone. Thank you for tuning in and joining us today. To all of our conference participants, this is our virology and vaccine research panel that we're hosting today. We have a lot of people on with us today who are uh, researchers here at UTMB at the Galveston National Laboratory and also the Sealy Institute for Vaccine Research. For all of you here today, I'm going to go ahead and read off your bios that we had for you posted on the website, and then I'll give y'all a chance to talk a little bit about what y'all do. We also have with us James Sowers, who is my co-chair for the panels. So first off, we have Dr. Pei-Yong Shi, who's the John Sealy Distinguished Chair in Innovations in Molecular Biology here at UTMB. His lab's research focuses on viruses that can cause significant human diseases such as dengue, Zika, West Nile, and SARS-CoV-2. They study mechanisms of viral replication to translate the knowledge into antiviral, vaccine, and diagnostic products. Many of his technologies have been licensed to leading pharmaceutical companies for countermeasure development. A recent example is his reporter neutralization assay that has enabled the rapid development of Pfizer's COVID-19 vaccine. We also have Scott Weaver, who's the director of the Institute for Human Infection and Immunity. He's also the scientific director of the Galveston National Laboratory and the chair of the Department of Microbiology and Immunology. Dr. Weaver currently holds the John Sealy Distinguished University Chair in Human Infections and Immunity. He has over 350 publications related to virology and vector biology and holds 9 patents for vaccine and diagnostic development for viral diseases. Next is Alan Barrett, who's our director for the UTMB Sealy Institute for Vaccine Sciences. He is also the John Sealy Distinguished University Chair in Vaccinology and the director of the World Health Organization Collaboration Center for Vaccine Research, Evaluation, and Training for Emerging Infectious Diseases. His lab works on developing vaccines against flaviviruses by studying virulence factors and working with live attenuated strains. We also have Mariano Garcia Blanco, who is an internationally recognized expert in RNA biology and virology and has been continuously funded by the NIH since the early 1990s. He has dedicated his career to studying how RNA protein interactions regulate gene expression in cells and viruses. These studies have focused on several areas of high biomedical importance, such as the intersection between RNA biology and host immunity. And finally, we have Dr. Richard Rupp, who's the Director of Clinical Trials and Clinical Research and is a member of the Executive Committee for the Sealy Institute for Vaccine Sciences. He leads the Sealy Institute for Vaccine Sciences Clinical Trials Group and is a principal investigator for clinical trials phase 1 through 4 in both pediatric and adult populations. Vaccines studied have included those protecting humans against cytomegalovirus, seasonal avian and pandemic influenza, pneumococcal disease, and human papillomavirus, dengue, varicella, HSV2, and meningococcal disease. So now I'd like to give the floor to each of y'all if you would like to tell us a little bit more about what it is that you do. Dr. Shi?
Thank you. Yeah, it's a great opportunity to join the panel to share our research and then to talk about the vaccine. We responded to COVID, and we're not used to work on the coronaviruses. As a virologist, we all feel obligated that we pitched towards in response to this pandemic. So what we've been doing is really not just only my lab, it's the community of the UTMB that we work together, and UTMB, we developed the first reverse genetic system that entitles us to manipulate the virus anywhere, any position we want. And that really enabled us not only for the translational medicine, but also the basic research. As you know, if time permits, we can talk a little bit about that. And that is very exciting. And then using those tools, we really shared with our scientific committee, again, through the UTMB community, like World Reference Center, that allows us to share reagents in real time with the different labs in the community, and then partnered with the Pfizer and other pharmaceutical companies to respond to this pandemic. That's what we did in the last year.
Thank you, sir. Dr. Weaver, would you like to tell us a little bit more about the work that you're currently doing?
Sure. So like Dr. Shi, I never worked on coronaviruses before February of 2020, but wanted to do everything we can to respond to the pandemic emergency. So some of the activities during the past year, Dr. Shi mentioned the World Reference Center for Emerging Viruses and Arboviruses. We were one of the first 5 laboratories in the US to have an isolate of the virus, and the first repository to provide viral RNA and the virus isolate to hundreds of labs around the country and the world to jumpstart their own diagnostic and research programs. In our own group, we've been working quite a bit on understanding how the variants that are appearing around the world are spread more efficiently. We do a lot of that work with Dr. Shi's group in animal models and models for human airway systems. We've also been screening a lot of samples coming into our hospital laboratories. These are the nasopharyngeal samples used to diagnose infection, to see which variants we have circulating in our region, to see which viruses are causing breakthrough infections of vaccinated people who are nevertheless becoming infected, and to look for other kinds of evidence that the virus is changing either to spread more efficiently or to become resistant to either vaccine-induced immunity or immunity after natural infection.
Thank you, sir. Dr. Barrett?
So the Sealy Institute for Vaccine Sciences decided not to work on new vaccine candidates just because academia is too slow, and I'll come back to that one in a minute. The clinical work we've done is going to be described by Rick Rupp later on, so I'm not going to say anything about that. The preclinical studies we've also been involved in, and that is the testing before they get into humans. And we've been involved in some of the studies taking monoclonal antibodies that will be used in humans and testing them in animal models to make sure they prevent clinical disease in the animals. And then they progressed on to go into clinical evaluation. And Dr. Rupp will talk more about that in a minute. The other big role that I've been involved in is something called CEPI, and that's a coalition for emergency preparedness innovations. And CEPI is making vaccines on the international level for preventing COVID, mostly in low and middle income countries. And I'm on the scientific advisory committee, and we got involved in doing COVID vaccines in the middle of January, and we kickstarted the Moderna vaccine, giving $1 million, and then before the NIH took it over. Subsequently, CEPI has been supporting 11 different vaccine candidates. Some of those haven't made it, but others have. And at the current time, CEPI is supporting 8 vaccine candidates. CureVac is messenger RNA, Novavax, Clover, biological E, and SK as protein, and the AstraZeneca chimp adenovirus vaccine. And what this means is, on the international level, they're milestone-driven projects. So it's not just a grant of $200 million. You have to go through milestones, and the milestones are evaluated by a group that I'm on. And the progress of those vaccines is being monitored as we go on. This is really a big issue for the world. So far, somewhere about 1 billion doses of vaccines have been made by the 14 approved vaccines, but only 59 million have been used in low and middle income countries. And that first started in February, and they're mostly the AstraZeneca vaccine. And there's a big need to get the vaccines out to these low and middle income countries. The plan is to get 3 billion doses out to low and middle income countries in the next 12 months, but the process, unfortunately, is going slow for a number of reasons internationally. So my big role has been involved in the milestone-driven production of those vaccines from preclinical, through clinical, and into manufacturing.
Thank you, Dr. Barrett. Dr. Garcia? You're on mute, Dr. Garcia. There you go.
No, it happens every time you come over 10,000 Zoom calls. It would be better, but no. So thank you, Danny and James, for doing this. This is really fantastic. And I'm glad you were able to rope some very, very good people from UTMB to do this. So I wear 2 hats in the university. I'm a professor first and foremost, and then I'm a chair also. So as a professor, I don't work with coronaviruses except small amounts of work in collaboration with Pei Young and Vineet. We are really interested in mechanisms. And as a professor, what I'm going to say now is I'm going to put a plug for how critically important fundamental science is to everything else that you talk about and you hear about in the newspapers. So it is really Pei Young's remarkable prowess in molecular biology that later on translates into Pfizer getting emergency use authorization very quickly. You could also argue another old UTMB person, Stan Lemon, and many others like, Charlie Rice at Rockefeller. Their very mechanistic studies on hepatitis C led to a cure for the disease. So what I do in my lab is try to understand disease process at the mechanistic level, and essentially work mostly on flaviviruses and really RNA related processes in the flaviviruses, and then in autoimmune diseases. And in the latter, I also have a company. I've started a few companies in my career, mostly driven by the 11 or 12 patents that we have out of academia. And there we're trying to use RNA therapeutics to treat autoimmune diseases. As a chair, what I tried to do first and foremost this year was make sure we were closed as little as possible. And the research enterprise continued for two reasons. Number one is, yes, you can see the firepower around this screen on coronaviruses. You didn't want UTMB to stop working ever on these things. But I also reminded the deans constantly that the other maladies that are affecting humans are still killing us during the pandemic. So we couldn't, I felt it was really critically important. So that was one of my major missions this year, is trying to help the deans reduce the amount of time that we were shut down and maintaining the research enterprise while trying to do it very safely with the expertise around this group. So that has been what's kept me busy for the last 12 months or so.
Thank you, sir. And finally, Dr. Rupp.
Hello. I think that we first got involved with this and the idea of doing the clinical trials probably in March. Pfizer reached out to us about doing the trials. We were told that we would not be able to do the phase 1 trials, so the first in human trials, but they were lining us up for 2 and 3. And the reason that we weren't going to be able to do these trials was because they were worried they couldn't get vaccine to us. They were worried that we couldn't get the labs, the blood that we're drawing and things back to them, which was a new thing that for us. We had participated in the H1N1 novel flu vaccine trials back during that pandemic, back in 2009. And after that pandemic, a couple of years later, the government decided that those trials didn't go fast enough, and we would have been in trouble if it would have been a nasty flu. A lot of people would have died. So we ran through a drill for a bird flu trial, and we put 175 people on in 3 weeks, very similar, surprisingly, to the COVID vaccine. It was a 2-shot deal, 3 weeks apart. We had to draw a lot of blood and learn how to move people back and forth and schedule. There's a lot of moving parts. And what happened this time that was shocking, that did not happen in the drill, is the bug was actually out there killing people. And so, the airlines were shut down, people weren't going to work anymore. And that's what Pfizer was trying to deal with. And they really couldn't transport things. And not surprisingly, things at UTMB started shutting down as well. They weren't letting us bring our people in for clinical trials because they were coming into healthcare facilities and they might carry COVID. We had to wear N95s and worry about all of that and keeping our staff safe when we were allowed to see people. And all those were complications that had not occurred in the drill. So that was a little bit surprising. But basically what happened was once they got past the phase one, we were one of many sites that this put people on as quickly as we could safely. We have continued doing the Pfizer messenger RNA vaccine for COVID, all sorts of, side things, giving people booster doses, we're testing the new formulations. They have a lyophilized or dehydrated one that we've been testing in people. Also a different dosing vial, and everything for vaccines has to be tested to show that it didn't get changed. We did the 16 to 18-year-olds, the 12 to 15-year-olds, and in the next week or so, we're going to be starting with young children. We also did the Moderna trial up on the mainland. They got started and going a lot later. We put over 80 people on, and we were one of the last sites activated. So it was a different trial. Even though the vaccines are so similar, the conduct of the trials was very different. There, as Dr. Barrett mentioned, we had also been lined up to do a vaccine for Merck. We were doing one that was using a measles vector. That's what was planned. It failed in early trials. We were also lined up to do the GSK vaccine that was in combination with Sanofi. That vaccine also did not fare well or do well in phase one, and I think they've gone back to the drawing board. So we've done a lot of listening and a lot of prepping for those things, but they didn't end up materializing. And I think right now the media, the public sort of think all the vaccines were a success, and they weren't. So anyway, that's basically what we've been doing during the pandemic.
Thank you, sir. And now I just kind of want to open the floor up and go in no particular order. But our first question we had is how have y'all had to change the work that you're doing in your labs in light of COVID-19? How has it changed your workflow? And I heard Dr. Garcia talk a little bit about his research isn't necessarily related to virus vaccines, but I'm sure that it's changed the way that you have to operate on a day-to-day basis.
Yeah, no, definitely. So I think because we're not coronavirologists and did not work, for example, all of the work on the autoimmune diseases was stopped for a while. I think some of the other leaders here know that I have a lab I had a lab in Singapore. It closed this February. Tears. I really enjoyed that lab. It was there, but they handled things quite differently. They did some things well. They did some things not as well as we've done it in the US. But I learned a lot from the way that National University of Singapore was using a team-based approach, so that instead of shutting things completely, although they eventually did shut completely, they brought teams. So I convinced the deans here that we should be able to have teams even for people not working in coronaviruses. Now that took a while. I wasn't as successful with that as I wanted, but yeah, we had to change the way we did our work. Some things got slowed down, other things actually moved faster.
Yeah, I think overall, just really as Dr. Garcia Blanco just mentioned, I'm very happy with the UTMB's operation in terms of keep the lab running. And for me, coming into the coronavirus, really, I don't have BSL-3 before, at the UTMB, but prior to the other institutions, I have done a lot of BSL-3 work. But over here, through very quickly collaborating with different colleagues, particularly Dr. Weaver, and we are very quickly can ramp up on whatever is needed can be done. That's really pretty amazing in my opinion. And then also coming from my nature, because it's really my nature based on my previous experience, I always strive to do team science. So whenever I come to a place, I always think about complementary expertise that we can work together, and then also think about like what ourselves, my lab can contribute. That's something other people cannot do in the world. I would, think about at that magnitude. And then really we can team up, and then wonderful things will happen when you have different disciplines and teams working together. And I think that's really the key to be successful in a very competitive landscape, right? And then of course, over the past year, I think everybody on this screen worked the intensities of what we have been going is unprecedented. Throughout my career, I really feel I've been always working hard, but the last year and a half is phenomenal. You just feel you have to, because it's an obligation that you have to in response to this magnitude of pandemic.
A couple of other factors we had working in our favor at UTMB are our high containment facilities. So we're kind of known for having BSL-4 labs because we're one of only 2 universities in the country that do that, but we have very extensive BSL-3 labs, and BSL-3 is the level required to do most work with the coronaviruses. And those labs were a little bit underutilized before COVID came along. So we were able to upgrade those labs thanks to some very generous donations from the Seeley and Smith Foundation, a local foundation here in Galveston, gave us millions of dollars to upgrade those labs, to put in some new state-of-the-art imaging equipment for animal research. And the other major advantage we had, unlike those of us on the program today, we have 3 other virologists who spent their whole careers working on coronaviruses. So Shinji Makino, Kent Seng, and Vineet Menachery. And those of us who started working on coronaviruses just last year, it was hugely beneficial for us to go to these experts and to get their collaboration, get them to help us design experiments. They're very familiar with the literature. If you weren't working on coronaviruses a year ago, you've been kind of drinking from a fire hose, trying to keep up with the scientific literature for the last year, but they were in a much better position at the beginning to do that. So that really allowed us, I think, to make some fast progress on a lot of different projects on our campus.
And also I would like to add, really, we at UTMB, we do have a lot of thought leaders who plays an important role at the global level, even at the policy. Like we just mentioned that Dr. Barrick sits on the WHO and all those panels. That really gives me at least a lot of insights of in terms of the future directions, the importance that will guide us to make the next move, which I think it's a huge, huge advantage and asset we have here.
Yeah, I'm gonna put a plug to Rick Rupp and the clinical side of UTMB, because I had this is going to be a little more personal. I was one of the first suspect cases for COVID because I flew from Italy, right? I made the huge mistake of traveling to Italy as the outbreak in Veneto was exploding in Milan. And it happened on the way back. I was in a business class surrounded with Milan businessmen who, was one of the epicenters in Italy. And then 5 days later, I developed a fever and blah, blah, blah. Long story short, it was not pneumonia. It was not COVID, but bottom line is I was one of the first patients in the emergency department protocol run by many clinicians. And it was impressive. And then I became actually a subject in the phase 3 trial that Rick, I even got my booster recently for in one of the 2-dose trial that Rick mentioned. And I think that's really something we need to really emphasize, is that Pfizer, BioNTech, places like UTMB have not stopped testing, right? We are still studying what happens so we can answer the questions that are really critical questions in combination with like Pei Young and others in the group. What is the correlation of protection? how are we going to do with new variants? How will immunity wane? All these things are still ongoing. So I want to put a plug in for the people who are doing those clinical studies today, still ongoing.
I think I'd add to that that everyone is being collegial. Everyone works together. There's no egos over it. Everybody has come together to do things. And Dr. Rupp and his clinical trials program, the first studies they did were not vaccines. They were actually a drug study for remdesivir and helping Dr. McClellan out. And so I think everybody's worked together. Interestingly, outside, you hear in the media about everybody is racing against each other and secrets, but the actual reality is everybody is collegial and works together. And as Pei-Yong said about the WHO and sepicals, it's quite interesting that the West Coast is starting 3 a.m. We start 5 a.m. It's in the afternoon in Europe and it's in the evening in Asia, and everybody gets on board to do these things. So in addition to doing your day job, you're also doing your COVID job. And everybody just gets on with it. It's very impressive. And I don't know how the West Coast people do it, get up at the 3 o'clock in the morning every time for these meetings that we have, and I just keep going. And the other thing, contrary to what the media says, all the farmers work together. They're all giving secrets to each other for manufacturing and production, contrary to what the media says. Everybody is working together. Everybody knows this is a public health problem that has to be beaten. There is no secrets between companies and this patent stuff. It's just, unfortunately, the media saying things. Everybody is working together. It really is very, very impressive.
That's great to hear that you don't always hear about that collaboration on the front page of the news, but that it is actually happening in the research world. So,
yep.
I mean, granted that we've had this larger scale global, push to create a coronavirus vaccine, how do you see this entire experience changing vaccine research in the future?
Well, I think for the first time, the mRNA vaccine has shown that can work. But of course, we anticipate not all of them will work if they put into that platform. But that really itself opens up unlimited possibilities in terms of vaccine, not just for infectious disease, but other indications. So I think this is a phenomenon and the potential is unlimited.
Yeah, this is the only thing I'm going to say because I'm not a vaccinologist, but I agree with Pei-Yong 100%. And I think that one cannot forget that even though these were the first that were widely used, that there was work for decades of people who made it possible, both in terms of knowing about the protein, but also knowing about mRNA technology, which, as an RNA biologist, it's incredibly gratifying that this is happening. But I agree with Pei-Yong. This is potentially a really important weapon in our arsenal from now on. Now I'm going to be quiet because I really don't know anything about vaccines.
I hate to sound negative, but actually there are 32 COVID messenger RNA vaccines in development, and they haven't all made it. And the reality is we all hear the headlines about the success stories, but not everyone is successful. I think it's been a phenomenal job by everybody coming together. I said collegiality, everybody's just got on with and done it, gone through, worked really hard. I think the difficult one here is the politicians and the media think that it can be done in a year every time now. And that unfortunately cannot be done. It's just not the bandwidth for people to continue at this pace indefinitely. Everybody's worked really, really hard. And I think it's a phenomenal what's been achieved, as Pei-Yong and Mariano were saying. But it just can't continue at this pace indefinitely. And unfortunately, that's what we're seeing from the media now and politicians is, it can be done in a year. And I was on a meeting yesterday for WHO, and this politician was saying, no, we know the scientists can do it in a year now. We know we can beat all these emerging diseases. It's not a problem what it used to be. It's just the political will to do it. And that isn't the case. Unfortunately, we're our own worst enemies. Unfortunately, being successful is that expectation now we can do it every time. Sorry to sound negative, guys.
So I'd just like to follow up on that that idea real quick. Kind of to my understanding and at the phase and training that I am in currently, a lot of what we hear is it takes $1 billion and it takes more than a decade to translate something at the basic science level, at the mechanistic level, to eventually get it into a clinical trial or some sort of human study. So I guess the first part of the question is, what do you think going forward would be a realistic timeline? Sorry to put you on the spot, Dr. Barrett, but what, is it less than 10 years now, or is it still 10 years is kind of the, what the kind of expectation should be? And then kind of to follow it up from there and kind of leaving the basic science and the clinical realm, what are some of the major challenges logistically with getting these vaccines to the rest of the world? Because it seems like the rollout in the United States has picked up its pace, but not necessarily in other parts of the world.
Well, a lot of questions there, James. I think it'd be good if Rick Rupp talked about the clinical stuff in a minute, about how difficult that is. And one of the issues is just squashing everything down, and everybody's expected to work all hours a day doing this. And Rick can tell you about him and his team, how much they've done to keep up with everything and what it's been like. The one thing we haven't talked about is the regulators. The regulators have also worked all hours a day approving things. And I don't know how the FDA have turned around the stuff they have, because they come out with these long reports within a week of evaluations, and they must work all hours a day to get that done. And that's the issue. Everybody's working all hours a day. Can it be done in a shorter time than we have now? You said 1 billion, I think it's over 2 billion in 10 years. I think yes, but a lot of it is driven by the money and availability of the money. And then for COVID, there was no limit on the money. And essentially, like for example, with CEPI, we've thrown 1.3 billion at 5 vaccines, and so that hasn't been a problem. And you get on and do it. I think that's the bigger issue, is the money required to do it. And in the case of COVID, Pfizer self-financed everything. I think it's amazing what they did. I bet they spent nearly 3 billion on what they've done in the last year and a half, and that takes a lot of resources to do that. And for some companies, it hasn't worked. And that's the other question, how you do that. On the worldwide stage, the problem is manufacturing and process. Regulators require at least 3 different lots made on different occasions to go through approval before a vaccine is approved, together with the seeds for the primary and secondary to do the manufacturing runs. And that's what holds things up. And can you find facilities? So everybody has said they were going to do manufacture vaccines on the expectation that only 1 or 2 would work. And of course, we have 14 approved at the moment, and there's just not the vaccine manufacturing capabilities around the world to keep up with everything. And that's slowing things down as well. And so it's the whole infrastructure that has problems from beginning to end. And one of the other big problems at the moment is we don't have many non-human primates left in the world. We've done so much work with COVID that there is very few animals left around to do studies. Lots of issues. But I think it'd be good if Rick talked about some of the clinical trials experience. Rick, if you don't mind.
Yeah, I think the big thing is basically money. Part of the reason why it takes 10 years to do it is because they have all these natural pauses built in. At the end of the phase 1, they look at all the data and, write the next protocol and make sure everything looks good before they invest any more money in it. And then you do the phase 2, and they're investing more money. It takes a couple of years to do it, and then they look at everything again very carefully before they move on to phase 3, because that's the expensive one. And if they move into that and they fail, they've just wasted a whole lot of money. And we did see a phase 3 vaccine fail very early in my clinic or career, which was the genital herpes vaccine by GSK. And it made it to phase 3. So part of all of this, is usually the caution. They don't want to throw their money away. Also, like the money was there for us to hire extra help, people coming in. Instead of having a week to put the data in, they wanted the data in within 24 hours. And then they had, 2 people on our butts all the time, with the company telling us, get the data in, you don't have this in. At all levels, there were a lot more people hired than usual. And, everybody's basically, I feel like, on us trying to make sure that we're doing it as fast as possible. And it's not the safety part that was skipped. All of that was done appropriately, but everything from like doing the contracts to, running the lab work, to inputting the data, to the statistical analysis, and all of that part was compressed. And I do have to say that I was really impressed with the FDA. When we did the pandemic drill a few years ago, they would not play with the rest of the government and refused to do that. And I thought they would do the same thing again, but they really came through and they really worked on having rules that worked and were appropriate, that they, it was this pretty incredible. I didn't think that that could happen. I thought it should happen, and it did happen. So I was impressed by all of that and continue to be impressed at how those things. I think some of those rules and the reworking of the rules will make things easier going into the future because they had to rewrite and do those things. I see Dr. Barrett smiling because regulators like rules. So I don't know. It might go back the old way. I don't know.
So Rick, the call I was on yesterday, Marian Gubler said she's not going to change. She's going back to those it was. COVID's extraordinary, and then it's back to normal.
Yeah, I think what Dr. Roop just mentioned is so important. And I think these messages needs to be stronger to the vaccine hesitancy population, because people still very skeptical, like we are just discussing, how fast this could be compared with the regular, procedures, right? And these informations, by taking a tremendous risk of compressing, not just the, one by one, waiting in a linear way, they are all staggered in the multiple, directions all at the same time. So I think that really needs to be out to let the mass population know that, so that we can remove at least a little bit on the vaccine hesitancy. And, coming from industry before, I always feel there are a couple of things to be successful. Of course, the will, the political will or the financial will, the money, that's important. The second is really the science, the approaches you use that needs to be right. The third, I always say, you need some luck, because it's happened to be this COVID. It seems to be, relatively compared with other pathogens for developing a vaccine. It's, I would say, yeah, think about HIV, how long we have been putting everybody's, all the intelligent, smart people have been on this for decades, right? So I think there are a certain level of a luck needs to be there, as always.
Dr. Shi alluded to this about the public hesitancy. And it seems like that's kind of one of the biggest barriers right now. And I'm sure that it's incredibly frustrating to spend all this time and energy and billions of dollars, like you're saying, developing this vaccine. And then the public doesn't want to take it because of something they read on Facebook or some rumor they heard from their cousin. So how do y'all think that now that we're at this point, how do we combat that? How do we combat the hesitancy from the public?
So Danny, let me just make a quick comment there. I was actually gonna bring this up, that you and James and medical students and people, you are in a very privileged position to help us with that. Okay, so it's very hard. If you read some of the social, the social science literature about convincing people to do things like, the whole campaign for seatbelts in this country, for example, things like that, and particularly vaccines in this day and age, it's very tough. Facts are not going to make it. We have to have the facts there. They have to be clear. But the majority of the people who don't want to get vaccines have their own facts. They have their own reality, and they talk to other people in that reality. So we're trying, actually, Scott has helped us tremendously with this. We have the American Society for Virology, and I'll send you the link to the videos that we've created. We've started a series of town halls. I think Scott has participated in many of them already. And we're targeting certain populations. You know, we are in tight community, this group, this task force that started this to try to not only put facts in, but to try to discuss with people. And one of the problems is we're a bunch of old people, right? So we need younger faces that go in there and talk to, because they're, for example, in the military, it's the young in the military who are very hesitant at this point. This is at least what they've told this guy, Dave Morenz has been talking to me a lot about this issue. And, you know, we need your help. So I'm going to give you guys the link for these town halls. Now they are letting postdocs, and I think they will let medical students co-host these with older people. We try to be as diverse a group as we can, but one of the big problems we have in our group when we talk to people is we don't have youth. So it would be great if you guys can get involved in this.
I think Mariano makes a great point. And just to begin with, I think if people see that this is sponsored by American Society for Virology, some of them probably never tune in at all just because it sounds too scientific to them. I think things sponsored by UTMB medical students or UTMB Department of Medicine or Family Medicine, those are more likely to get some of the folks who are on the fence or against getting vaccinated at this point. I think the other thing, back to the general discussion about where we are with vaccine development, progress has been great, and we're seeing most places in the country, the number of cases coming down, fatalities coming down, hospitals having plenty of capacity yet, but this is not over. And I think we're entering a period with a different kind of risk coming along. And that is, if we have, say, half of the people in the United States with immunity, that's enough to slow things down, but not to stop spread completely. And so we're probably entering a time now when the virus is going to have a lot of selective pressure on it to find ways to overcome our immunity. So somebody who's infected, if they contact 4 people, 2 of those people are going to be susceptible and they'll become infected and the virus will do well. But 2 of those people are going to have some degree of immunity. But if the virus finds a mutation or a recombination site or other ways to counteract that immunity and infect those people anyway, we're going to have the virus changing very quickly to circumvent our immunity. And that's going to be a very risky situation. So we really need to get vaccination to the point where these viruses are barely circulating at all, not only in the US, but in other places around the world, because any mutant anywhere arrives on our doorstep very quickly. And so we need to get herd immunity up, and we need to suppress the ability of the virus to find new mutations and new ways to counteract immunity.
Go ahead.
I just wanted to say, I mean, the vaccine hesitancy isn't really anything new. It may be worse now, but a lot of it just escapes common sense. You know, they're seeing all these other people being vaccinated and they're doing fine, and yet somehow feel that the vaccine is so risky for them. That's been one of the biggest disappointments in my life. The first study I was principal investigator was on the HPV vaccine, Gardasil. And before that, I had done some acceptance work on that. You know, would people accept it? Would they take it? And the vaccine's a home run in the vaccine world, you know, and we're still struggling to get teens vaccinated with it in this country, which is this stupid. You know, so much data and so much information. And, you know, I'm shocked. You know, I was 2006, so we're well over a decade and we're barely making any headway in it. Although this talking about youth, I've heard that the medical students went out and talked to people and vaccinated 100% of the people that they talked to. And somebody had asked me, well, why was that? And I think, oh, is this the confidence and the energy that, swayed people to do it? Because we don't see vaccination rates anywhere close to that in our clinics. So anyway.
If I can just add to that one, I think locally, UTMB has done a great job in Galveston County with immunizations. And that's because it's gone out into the community to give the vaccines, just as Rick said, the medical students have. One of the issues, I think, for the United States is the healthcare system is built around people going to the hospitals, not the hospitals going to the people. And I think we could reduce hesitancy if we went out more into the community to get people vaccinated. It works very well, as the medical students have shown, and as UTMB has shown, immunizing in Galveston County. And other areas in this country don't do that. And if you look at the United Kingdom, where they opened stadia all over the place, they took the vaccines out, they got a very good rate. And interestingly, to follow up what Scott was saying, the UK has shown that the variants are all produced in the people not vaccinated because the variants are allowed to produce, particularly in children. And I think that's something we don't really get much to understand in the United States. The variants appear mysteriously in this country. We don't really talk about their origins and how they're generated. And I think that's something that we could do to help hesitancy if people understood that they're essentially incubators, they're not vaccinated, to produce these variants.
And it sounds like the next phase is kind of where Dr. Rupp specializes in the adolescent population. We just started vaccinating people down to the age of 12, I believe, here in Galveston County or in the state of Texas. So that in particular, I feel like that's going to be one of the more difficult populations to try and get vaccinated because they can't drive themselves to a vaccine clinic. And they also might have overbearing parents who are afraid of some kind of side effects affecting their child's development. So what do you think, Dr. Rupp? You think that this is going to be particularly difficult to get kids vaccinated?
Well, I think it's going to fall along the lines of their parents and what their parents' thoughts are about all of that. A lot of people don't think there's much risk to their kids, and so they won't get vaccinated. But as indicated, the biggest reason why people don't get vaccinated is basically that they have to go out of their way to get vaccinated. And so there's a lot of work on vaccinating the kids in school and out away from UTMB, and they'll be easier to reach. Myself, I'm medical director for the school-based clinics on the island, and we're getting a delivery of the Pfizer vaccine this coming week. And so we'll start vaccinating that group. We have permission slips that have been put online for the parents to fill out if they'll allow us to vaccinate their kid without them present. So we're trying to overcome that. We'll be looking for kids all summer and dragging them down and giving them a shot. One of the nice things is that there's so many more vaccines now that the kids aren't quite as afraid of needles as they used to be. So it used to be that your last shot was either when you were 4 or 6, and then you got your tetanus shot when you were 14. Oh my God, they were terrified of that. The needles grew so big in their minds and so much fear, we'd have them kicking and screaming. But nowadays the kids get more shots and they're a little bit more used to it than they were. So just an observation.
So I'd like to switch gears a little bit and kind of talk about how the technology that's been developed over this past year, and it's been alluded to a little bit in earlier discussions by Dr. Garcia Blanco with some of the research that he's doing on autoimmunity. But I'm wondering how some of these technologies that have been developed in this past year, kind of in response to COVID, might be then utilized in other areas of human health going forward?
Well, I think the mRNA technology that got developed in many different labs, right? For example, if you look at the history of Moderna, they really started doing therapeutics. They would sort of be an alternative to gene therapy, right? Expressing proteins for therapeutic treatment. Those programs, many led by a very good friend of mine, Melissa Moore, who's CSO of that area of Moderna, are still ongoing, actually. So, I think there has been now there are enormous challenges to, and as Alan said, although I would disagree with Alan in the sense that 2 out of 32 is not a bad number for some of this very high risk research. But there are enormous challenges to take this to the next level. There has been a tidal wave of new companies. Every day you hear of a new one. Orna, a company that came out of Anderson's group at MIT, making circular RNAs and now translate proteins much more efficiently than the linear RNAs do. They last a lot longer. We actually worked on these things back in the 90s, but that was the caveman age of RNA therapeutics. So I am very, very, I'm an optimist by nature. So I think that these technologies will revolutionize medicine, perhaps not in your lifetime, but maybe the next generation of physicians will actually have a whole new tool set to work with, not to mention diagnostics. So I think that one thing, the last thing I'm going to say is that we actually cannot forget that these pandemics, that I think I heard a very good talk by Scott the other day where he has these circles of what has happened, and he asked what is common about all these things. And one of the things, everybody looks at the world a little bit differently. For me, one of the commonalities, these are the last few have been that really have made, you take HIV, influenza, COVID, dengue, Zika, they're all RNA viruses. So we need to learn a lot more about how these machines work. And obviously a little bit self-serving, given that I'm an RNA biologist, but I'm very, very optimistic that we should ride this optimism and really convince society it's time to invest in understanding these things better.
So kind of a final question.
Just to add something to James. The one area we haven't talked about is the adjuvants. And that's a real area where there are opportunities to make improvements, that enhancing the immune response. We're still using the basic 5 or 6 adjuvants that we have for many years. And there's a lot of research in adjuvants, but they haven't really made prime time yet. And I think when those adjuvants make prime time, then I think we can see some big changes in the way that we do vaccinations, because they will give much better immune responses. There's some in development, but they're a bit reactogenic and they're not appropriate for use in humans. But I think that's a big technology area that probably will make a big difference in the next few years to enhance the vaccines that we have around now, as well as the new ones coming along.
That's good. And so we're kind of coming to the end of our time. I wanted to ask one more question to sort of wrap things up. So granted that, the world is going to continue to grow, population is going to continue to increase, the globalization is going to continue to expand. I personally think that there's a very real possibility that something like this could happen again in the future. And so I want to know what y'all think. Do y'all think that this experience was a good learning experience and that we'll be able to respond more quickly, that we'll be more prepared in the future? Or do you think that it's going to be just as much of a nightmare if it did happen again?
I think, Danny, it could have been much worse. If you think about just coronaviruses, the first SARS outbreak involved a virus that had about 10% case fatality rate. It was not as transmissible, or we would have had a pandemic that could have been much worse. Probably came very close, though, to going pandemic at that time. So we've known for for 18 years that first SARS coronavirus was not the first that would come our way. And nevertheless, coronavirus research waned during that time period. Vaccine development pretty much came to a standstill. One thing that I think we have been doing better recently is more surveillance projects to identify these viruses that are risks to us around the world. And I think that USAID, for example, is putting out a new program already to do a lot more work on identifying viruses in all kinds of hosts around the world to help to at least set the stage for looking what may be risky or what may not. But I think we need to do more in areas of the world where these viruses tend to emerge. We can still improve vaccine development, and that'll be helpful, but many of these outbreaks are going to be too fast for a vaccine. If this had been, let's say, Zika virus, which came our way about 5 years earlier, these vaccines would not have been available in time to really make much of an impact on the trajectory of the outbreak. It was much faster than SARS-CoV-2. And so I don't think we can expect that vaccines are always going to be our solution to these problems. We have to be able to anticipate them earlier, find the risks before the movement into the human population occurs, and figure out ways to mitigate those risks.
I would agree with Scott. I think the problem's multiple levels, not only surveillance, but also what we call plug and play for vaccine development. So organizations like Gates, WHO, NIH, CEPI are all interested in this idea of technologies where you can just store vaccines to control outbreaks when they come out. And what's called the blueprint diseases by WHO, there's this number of diseases you anticipate, and of course, what is called disease X, and I think SARS-CoV-2 is what we would call disease X. Everybody after the Ebola problem in Africa in the mid-2010s has been looking at disease X. How do we get ready to prepare for the unknown? And it's everything has to be put together to do that. The one area I think everyone is forgetting at the moment is that as soon as you have a large outbreak, epidemic, pandemic, people don't take the normal vaccines. And a great example is Shingrix vaccine. The sales have gone down 50% this year because people aren't going to get healthcare, they're staying at home. And so the normal vaccines that we give out to stop these diseases that we think we've conquered are going to come back. And measles has become a big problem in Africa now since the Ebola epidemic in 2014, 15, 16, because all healthcare workers were dying from Ebola. They didn't want to go out into the field. And so normal vaccines weren't given. And I think it's a really complicated scenario at many levels that we have to look at moving forward. But I think it's pretty clear, as Scott and others have indicated, that we're going to get another of these viruses that's going to jump out again in the future. And the question is, will we be prepared? And I think Scott's right that we won't be prepared, unfortunately.
Yeah, you know, there's a famous phrase of Jorge Santayana, right? Those who don't remember history are condemned to repeat it. And I think this is a great time to remind people about this over and over again. And it's not only vaccines, it's not only, I mean, each of us has to do what we do well. And I think people like me, if we can contribute, people like Pei Young, you know, we have to make sure that we continue studying the molecular mechanisms of these different families of viruses so that we can at least anticipate a potential antiviral, a potential, or not antiviral, maybe something that dampens the host response a little bit so we don't kill ourselves with inflammation. So I think we have to, you know, we cannot forget this. It's not that it may happen. I think there's no question that it's going to happen. The question is, and we have a good series of candidates of what the, you know, is it going to be a nuvovirus? Is it going to be a corona? Is it going to be an influenza? Yes, the answer is probably yes. It's probably going to be one of those. So we should work on them, not to even mention filoviruses and others. But Pei Young, I know you've been thinking a lot about this, so I don't know if you.
Well, I think, yeah, it's a no-brainer. The next thing will come. It's a matter of how we put our acts together to be prepared for the next one.
That's great. And it's given me a lot of confidence hearing from all of y'all today. Like, seriously, it sounds like y'all are very busy people. I appreciate y'all taking time out today to speak to us. If anybody has any closing remarks that they'd like to make.
Just thanking you guys for doing this. I think it's a great idea. Let's do it again.
This is great. Absolutely. Yes, sir. Next time that there's an epidemic, we'll get you all together.
Let's not wait. Let's do it beforehand. Let's talk about anticipation.
Yes, sir. Well, great. Again,
Dr. Garcia Blanco, could you please provide that link in the chat or to one of us, and we'll get it circulating.
Absolutely. I'll do it right now.
Great. Well, thank you all for spending your time here today with us on Saturday. And we're going to go ahead and wrap up our session. And for our conference participants out there watching, I hope that you learned something valuable today. And also our next session will start at 1 p.m. Thank you.